Tell me if any of this works. Oh,
Yeah,
okay.
okay,
Now it's
that worked.
working.
There we go.
So video recording.
Woo! Yes.
Wonderful. So I'll go into presenter mode. Hello, everyone. Welcome back to week 2. This week, hopefully, it's not going to go for as long. It should be around an hour, if not less. It's more of a focus on biochemistry today. So you've kind of really started the harder stuff now, which is good going. And yeah, we'll just work through it. So we've got some biochemistry, physiology, and HKS. So to begin with, we're going to start with central dogma, which in essence is just saying that you have DNA, the DNA can be replicated. If you want to create protein, the DNA should be transcribed to RNA, and then that RNA is translated into protein. Initially, it was thought that this is a unidirectional one-way process. That's now no longer true. There's some viruses, for example, the HIV virus, who has enzymes such as reverse transcriptase, which can get the RNA and work backwards and get it into DNA, as well as there's what we'll talk about later is alternative extron splicing, which is an indirect pathway to this. So the central dogma, ironically, is not now completely factual, but in essence, it's mostly true. So these are some important essential functions of DNA. So the first, I should just say, we'll put these up in our drive after this. So the first, maybe we can put it up now, maybe someone can put it up while I'm talking. So the first function of DNA is to store genetic material, and essentially a strong sugar phosphate backbone with complementary nucleotides facilitate this. It also preserves genetic material, so it's quite robust and should be able to survive in altered homeostatic states. And yeah, it can also replicate, which is important for cell mitosis and cell division, which is important for healing as well as growth. And mutation is also important because mutation is essential for survival of species. And yeah, so DNA polymerase facilitates the replication through promoters and initiating sequences, and mutations can occur through the complementary nucleotides. The structure of DNA. So DNA is a long polymeric molecule made up of subunits called nucleotides. Now, this is high yield, so remember this, nucleotides consist of deoxyribose sugars, phosphate groups, and nitrogenous bases. The difference between DNA and RNA is that DNA is double-stranded. It has a phosphate group and thymine, whereas RNA is single-stranded. It has uracil, and it has ribose instead of deoxyribose. DNA is double-stranded, as said before, and it has an anti-parallel arrangement, which means if you go from the base to the top on one side, it's in a 5' to 3' direction, and a 3' to 5' on the other side. It's important to know that purines are adenine and guanine, and pyrimidines are cytosol and thymine, and there's also uracil. So that is, for some reason, they might ask a question on that. And it's important to remember that A binds with T and C binds with G, and the binding is determined by the hydrogen bonding. You can see that in the image. Can you see my mouse?
Yeah, we can.
Yeah, okay, so that's determined by the image in the top right here. You can see the hydrogen bonding, facilitating that relationship. So DNA replication, these are all the steps. I wrote this in mind that you'll come back to this to like copy into your notes. So yeah, so first step is that you'll have something called DNA helicase, which will denature DNA at an origin of replication. And that will facilitate the appropriate enzymes coming in to replicate it. There's something called topoisomerase, which unwinds DNA. So once you've split the DNA helicase and it splits it up, there's supercoiling going on, and topoisomerase is important to kind of mend the coiling and provide stability. And SSBs bind to unwound strands to stabilize them and keep them apart. On the lead-in strand, you have an enzyme called a primase, which inserts a primer, and DNA polymerase will attach to this primer and then move into a 5' to 3' direction, adding complementary base pairs. On the lagging strand, I'll explain further what a lead-in and lagging strand are in the next slide. Maybe we should do that first. So you can see that because it goes in a 5' to 3' direction, on one strand, it's just going to be continuous because the DNA segments will be added in that direction. On the other strand, because of the anti-parallel nature of DNA, you can see here this is a 3' to 5', but it has to go in the 5' to 3' direction. So it'll lag behind a bit and it'll be discontinuous. So on the lagging strand, RNase primase will synthesize primer as before. However, the movement of DNA polymerase from 5' to 3' direction will result in discontinuous strands of DNA, which are called Okazaki fragments. Eventually, RNase H will remove the primers, and DNA polymerase 1, which is different from DNA polymerase 3, used in DNA replication, used before, will replace the RNA primer with a DNA strand, and then a ligase, something called DNA ligase, will seal the DNA fragments to make the strand completely continuous. Because before the ligase, there's even though there's DNA in between being inserted where the RNA primer was, they're still not connected and bonded. So you need the ligase to seal the bonds, and those fill in the gaps. This is just some information if you're interested. It's low yield.
David, I just jump in here. So might be a bit more obvious if you like drew on here. So on like this bottom fragment, like because you're going from 3' to 5' to 3', it sort of just works. Like you just go this way, but on the other direction, on the top strand, you need to go the other way. Like the new strand that's being synthesized to follow the 5' to 3' needs to go the other way. So it's actually heading in this direction. It's heading in the opposite direction. But because like you're still waiting for this bit to get unwound, it can only really do like the replication in segments. So it might do a little bit here. It might start here, do a little bit replication here, and then another primer will come, attach here, and then you do this segment, and then another primer will come here, and then you do another segment. But in this case, in this bottom one, because you're just going in the straight direction, you don't need to really wait. You can just keep going straight, if that makes a bit of sense, hopefully.
Yeah, beautiful. And if you have any questions, just pop in the chat and someone will answer it. And yeah, so DNA, a buzzword to remember is DNA is semi-conservative. So one parent strand will be in each replicated strand that's preceded. So as you can see, this will be the original parent strand, and then the helicase separated that from the other parent strand, but there is one parent strand per new complex of DNA that's created. Telomeres and telomerase. So what you'll realize is at the end of the construction of the lagging strand, you'll have, I'm going to copy Andrew and do some annotating. Chat popped up. Okay, there we go. You'll have some primer here, and then the primer will disappear. So you'll just have a bit left over of this continuous strand because it's gone through the process of ligase, etc. And if you keep replicating the DNA, you're going to keep having these little sections of no DNA, and the strand will become shorter. So in order to prevent that, let us get rid of these annotations. In order to prevent that, you have something called telomeres. So because of the 3' overhand in the telomere and that each replication, the DNA will get smaller, a RNA template will go up to the 3' overhand and extend the parent strand, as you can see in this image. Then DNA polymerase will act over here and will fill in the gap. And that will extend the length of the DNA. And it's important that this is essential in prolonging life of the DNA. Telomeres are closely associated with the aging process. And when you do cancer, they're important because they lead to the immortality of cancer. So what happens if mistakes happen? And how do mutations occur? Sorry, my charger fell. How do mutations happen? So one step is as proofreading, that is an ongoing process during replication, in which DNA polymerase will correct errors in the 3' to 5' direction while it moves in the 5' to 3' direction. There's a mismatch repair that occurs shortly after replication, and excision repair that is continuous and repairing nucleotides that are damaged. So you can see up here is the proofreading that's ongoing with DNA polymerase. You can see a mismatch repair that occurs shortly afterwards, in which these several base pairs are taken out and replaced with the correct pairs by the DNA polymerase 1, which is different to the DNA polymerase 3 in this example. And then you also have excision pair, which is a continuous process. And it usually happens if there's chemical or ultraviolet radiation. So next, we're moving on to the second stage of the central dogma, which is transcription, which involves initiation, elongation, and termination. So the template strand is used as a reference point for a pre-mRNA strand that will be synthesized. It's quite similar steps to the DNA replication, which can be confusing, but hopefully if we break it down in steps, you'll be able to see that. So RNA polymerase will bind to a promoter sequence, and transcription factors may assist with this. The template strand and the promoter sequence will determine the direction of movement of the RNA polymerase. Transcription factors and other factors may bind downstream or in other locations not closely associated to where the RNA binds, and conformational changes will occur. We'll show you a picture of this later on. And there's also enhancers and repressors along the DNA strand that allow for the modulation of the transcription of certain proteins, etc. RNA is synthesized in the 5' to 3' direction as well, and it is also an anti-parallel. However, remember that it's read in the 3' to 5' direction. So sometimes I'll ask you that. Read in 3' to 5', transcribed into the 5' to 3' direction. And there are base sequences in the DNA that specify the termination site. So here are some nice pictures we found. This is a promoter where the RNA polymerase will latch onto, but there's some enhancers and other protein structures that facilitate the conformational change of the DNA strand to facilitate the transcription process carried out by RNA polymerase. Here's another nice picture just to show you the function of enhancers and other transcription factors that support the process. And yeah, there's another you can go through in your own time, but we've kind of gone through the steps, and we'll go through the post-transcriptional modulation in a second. So the products of transcription, there are 3 types of RNA: messenger RNA that codes for proteins, ribosomal RNA that codes for ribosomes, and transfer RNA or tRNA that carries amino acids to ribosomes to decode mRNA into a protein. It's important that prokaryotes only have one RNA that does all of this, whereas eukaryotes have different types of polymerases to create these different structures. Sorry, prokaryotes only have one RNA polymerase to do all 3 structures. And yeah, it's not proofread, so there's about errors every 10 to 4 to 10 to 5 base pairs. It's quite prone to errors. Splicing, dicing, and processing. So at the end of your pre-mRNA transcription, you'll have some areas of coding that aren't actually involved in the translation or conversion into protein. These non-coding structures are called introns. I think of it as they stay inside, and they never come out to become expressed. And they're spliced out by these structures called spliceosomes that will, through conformational manipulation, can put stress on the strand and take away the introns while stitching together the extrons. The SNRMPs, which you can see in this image as well, these little structures. The spliceosome is a complement of these 2 together, but the SNRMP will also stitch them, the extrons together. Extrons are these green bits, and these are the coding parts of the DNA, of the RNA, sorry, these are the coding parts of the mRNA. There's further processing to ensure the stability, and this all occurs in the nucleus. So remember that. So methyl G cap is added to the 5' end, while a poly A tail is added to the 3' end. And this supports stability. So think of, like, those plastic tips on shoelaces that prevent them from unwinding. That's the function of the poly A tail. And yeah, these support the stability. There's other functions as well, but that's not important, or you won't be assessed in those. Now translation. So we get into the final part of central dogma, which is the creation of proteins that are actually functional and do things like become enzymes. So you have a codon on the mRNA strand. So if we go back to here, there's each 3 nucleotide base pairs will create a codon. And these codons can be read and associated to amino acids. So you have these, we'll talk about them in a bit, but these structures called tRNAs, and they carry amino acids. They will read the codons on the mRNA and will then attach the associated amino acids appropriately. We'll go through this in more detail in a second. So this table is for the codons. They're read in a 5' to 3' direction, And as you can see here, there's actually some redundancy. So several 3-letter pairs code for the same amino acids, which is important because often this last third pair is called a wobble pair because there can be mutations or alterations to this. However, the end product does not change and it is still functionally intact. So it's important to remember that while there is redundancy, there is no ambiguity. So no 3 pairs code for 2 amino acids. However, several codes code for 1 amino acid. I hope that makes sense. Tell me if it doesn't. But yeah, only 1 triplet codes for 1 amino acid. Not one. There isn't a triplet that codes for more than 1. That's not possible. But there are several triplets that are unique to 1 amino acid. I hope that makes sense. So now let's talk about ribosomes. In prokaryotes, transcription and translation occur simultaneously, whereas in eukaryotes, they're separated by space and time. For example, transcription will occur in the nucleus, whereas translation can occur in rough endoplasmic reticulum or in the cytoplasm. So there are 2 subunits of a ribosome, a small 40S and large 60S unit that together constitute an 80S ribosome. And these are unique to eukaryotes. They dissociate when not translated, but we'll get to this in a second. And the ribosomes are also made up of rRNA and proteins, which we've spoken about before. And these are bound by ionic and hydrophobic bonds. So it's important these are not covalent bonds and they do dissociate. Ribosomes are not specific. They can acquire any mRNA and attract all tRNA. We'll talk about tRNA more in a second. So if you look here at the ribosome, there's also 3 sites that are very important for you to understand. So the A site stands for amino acyl. You can think of it as amino acid as well. And this is where a tRNA, which is the structure that carries an amino acid, will bind to the mRNA codon and read it. So if the mRNA is over here, it's reading it from here. You can't see my mouse here. I asked that, I think. I'm going to assume you can. You then have a P site, which is, you can think of it as peptidyl, and that refers to the peptidyl transferase, which is the enzyme used to add amino acids to a growing polypeptide chain. We have a picture of this in a second, but if you think of it, say you have several amino acids here, you have a structure bringing an amino acid here, a peptidyl transferase bond will occur. There. There's a nice image coming later on. E is where an unbound tRNA, so once it's given its amino acid to the polypeptide, goes before leaving the complex. And the important word to understand is translocation, which just refers to the cycle movement of tRNA going to the next site. I realize I haven't fully explained what tRNAs are. I will in the next slide, I think, but they're just little structures that carry amino acids. They give away the amino acid, and then they leave the structure. tRNA, here we go. tRNAs have an adaptive function by binding amino acids, mRNA, and ribosomes. Their conformation or shape facilitates this. Amino acyl tRNA synthetase charges tRNA with an amino acid, which involves the use of ATP. So over here you can see the amino acid, here you can see the tRNA, and an enzyme called amino acyl tRNA synthetase covalently put these together. GTP, it's important to know GTP, it's not so important, but the as opposed to ATP, GTP is consumed in the delivery of tRNA to the A site, or like the entrance site translocation. These are the steps of translation. Again, these are more orientated for your notes than for the presentation, but mRNA will bind to a small subunit with a charged tRNA anticodon attaching to complementary antiparallel in a complementary antiparallel direction to mRNA. First is AUG methionine. So this is always the starting one, and sometimes it's cleaved off afterwards. tRNA then attracts the largest subunit, forming the initiation complex. Next tRNA complex has a codon-anticodon interaction at the A site, where peptidyl transferase covalently forms a peptide bond with amino acid subunits. The polypeptide growing will be from the N-terminus towards the C-terminus. Again, there's an image of this later on, so just remember to come back to this. First tRNA complex translocates, which just means it shifts over to the site P, and then exits site E. New tRNA without amino acids will move to site P as the original one moves to site E. Again, all that's saying is that there will be original one in the site A that will move to the site P, and so a new one will go to site A, and it just continues to cycle. When a stop codon is read, a release factor binds and a polypeptide is released, which ends translation. Here's the picture that I've been talking about the whole time. So you can see there is a tRNA with a growing polypeptide chain associated. And we have a newly bound charged tRNA come into the A site. A bond will form like this. The peptide then will shift over here. And the tRNA in this spot will become sort of naked. The translocation will then occur in which these both shift forwards. As the 3 goes into the E site, it'll be ejected, whereas the 4 now has that growing polypeptide chain, and the process continues. Clear all drawings. Okay, almost done. Last 2 slides, post-translational modification. So this is after you've modified, you've created this polypeptide, how does it become functional? So there's 3 things that can occur. Either it will be transferred to a cellular location, which will determine its function. It'll have to fold to be conformationally active, which is important for enzymes. And you also regulate protein activity through other processes, which we'll talk about in a second. So polypeptides will fold in 3D conformational shapes. Some will do this spontaneously, others may require chaperone proteins. Further modifications, such as glycolization or phosphorylation, occur to enhance signaling or charge the complex. This may occur in the endoplasmic reticulum and then go to the Golgi complex, where it's packaged and shipped through vesicles to maybe to the plasma membrane or to a particular site in the cytoplasm where it's required. And the shipping determines whether it's stored so it can be sequestered or whether it's transported to a cellular location. Some proteins need to be activated through proteolysis, which is just the breakdown of protein, for example, proinsulin to insulin. So insulin is only activated once some of it is cut off, and then it becomes active. And also, cellular signals can control maybe there's too much protein being produced. So either it can interfere with those enhanced repressors we were talking about for the DNA, or it can break down protein by something called ubiquitin. Okay, now we're going to talk about cellular energetics. This is quite low yield and hopefully we can get through quite quickly. If there's any questions, you can let me know. And guys, if you look at the questions while you're answering them and you need help, just ask me. I'm just looking at the chat. Okay, Andrew's on it. Awesome. So thanks, Andrew. Cellular energetics. Okay, give me 1 second. Let me just see if I can see this question. Chats. Sorry, almost done. Okay. Sorry. Do you have to know the process of protein structure, primary structure, secondary? Yes, you definitely do. I'm about to talk about it. I think it's maybe the second slide, but yeah, you do have to know it. It's very unlikely to be examined, but it is background to understand what an enzyme is and how protein goes from this one strand that's being translated into something functional. So we'll get to that in a second. How do I go back? Let's just go back to here. Sorry about that. Let's start. Quick protein is exactly what you've just asked. So quick protein recap. Polypeptides are polymers of amino acids, in which amino acids are the subunit and covalently bounded to each other by peptide bonds to create a chain. Proteins consist of one or more polypeptides. Who cares? The conformational protein determines its structure. So hopefully you remember this from year 12 or your equivalent, but you have a primary structure, which just refers to the ordered sequence of amino acids bonded by peptide linkages. You have secondary structures, which is a stable arrangement of amino acids by hydrogen bonds, and these refer to the helices or beta sheets primarily. You have a tertiary structure, which involves a 3-dimensional folding of polypeptide bonds and involves other forces such as hydrogen bonds, ionic bonds, and dispersion forces. And then you have a quaternary structure, which is the arrangement of 2 or more folded polypeptides to create a functional unit. There's mostly hydrogen and dispersion forces between these subunits. What is an enzyme? I know you all know this, so we'll go through this quite quickly. An enzyme facilitates a chemical reaction by lowering the activation energy pathway by providing an alternative reaction pathway. So it increases the rate of reaction, but it does not influence or affect the equilibrium constant or yield. Substrates bind to an active site on an enzyme, and the enzyme then will modify its conformation to accommodate the substrates and facilitate their reaction. An enzyme is not altered by a reaction, and an enzyme, again, this is like a good conceptual understanding. Another way to think about it is an enzyme increases the rate of reaction by lowering the activation energy. And by lowering an activation energy, a greater proportion of reactions will become successful. There's more substrates that are viable to react successfully. And this will increase the rate of reaction as a greater amount of product is formed per unit time. At low concentrations, an enzyme greatly increases the rate of reaction. At high concentrations, enzymes may become saturated. And at that point, the rate of reaction will reach a maximum and plateau. Without an enzyme, the rate of reaction will increase steadily as substrate concentration increases if you're putting more substrates in. Some laws of thermodynamics, which are not super important, but maybe they'll help you conceptually. The first law of thermodynamics is energy cannot be created or destroyed. It is just transferred through processes. And the second law is that entropy increases in nature. So this is quite a difficult concept, so I rewrote it a few different ways. So entropy increases in nature essentially means there's a movement towards disorder. So things will diffuse away from each other or they'll move away, etc. So during energy transformation, some energy is lost to disorder, which is a difficult concept and it's not super important you understand, but maybe it could help you. No energy, this means that no energy transformation is completely efficient in an open system. So I think you've had these lectures. And these are some concepts that I didn't understand, and I spent lots of time researching. I don't know if you want me to go through it now, and it might be better to read through it by yourself. But essentially, you'll see change in G a lot, and all change in G refers to is the energy capable of doing work during reaction, or capable of doing something. And this is known as free energy. The other factors are defined for your interest, if that interests you. A reaction is spontaneous if G is negative, and free energy will then be released in the process. Yeah, again, for your interest, some of this is quite interesting, but it's not very high yield. If G is less than 0, reaction releases energy, and this is an exergonic reaction. G is greater than 0, reaction requires energy, and it is endergonic. G equals 0, reaction is at equilibrium. Do tell me if you want me to explain it more. So I was confused because in year 12, we learned something different. We were told that if H is positive, the reaction is not spontaneous, and this is not true. So again, this is going more in detail why what we were learned in year 12 is incomplete and some examples to illustrate it. I think it should be quite clear. So if you have any questions, hit me up, DM me, but I think we might skip it just for the interest of being relevant. Chemical reactions. So generally, atoms are reorganized by breaking pre-existing bonds and forming new ones. This may lead to a net release or require energy. What is metabolism? Metabolism is a chemical reaction that occurs in order to sustain and support the life of cells, tissues, and organisms. If that change in G stuff confused you, just ignore it. If it interests you and it helps your understanding, then you can read it. But I'm also scared of just confusing people and making them scared. So yeah, don't worry if you don't get it. If you want to get it more, you can contact me, but don't stress. So again, a chemical reaction occurs in order to sustain life and support life of cells, tissues, and organisms. So you can see how metabolism involves very intimately chemical reactions. What is energy? Energy can either be potential, which means it's stored, for example, in chemical bonds, or it can be kinetic, which is expressed through movement. Again, as we've established, energy can be converted, but it's not 100% efficient. So while according to the first law of thermodynamics, energy is not lost, according to the second, in an open system, it is not 100% efficient. Some other key terms and concepts to understand. Catabolism, think of it as a catastrophe, refers to the breakdown of a macromolecule. This involves oxidation and releases energy. In the body, often nutrients are broken down in order to extract energy, and useful energy is stored in chemical bonds in ATP to be used in other reactions and can be coupled to anabolic reactions, which are the synthesis of macromolecules from precursors. It typically involves reduction reactions and requires energy. It often requires ATP and will convert ATP to ADP. More on entropy if you're interested, not super important. This is a nice diagram that demonstrates the kind of the coupling between anabolism and catabolism, in which the breakdown of macromolecules will have an end product and kinetic energy, but it can also create these little coenzymes that facilitate other reactions. So they acquire some of that energy, such as in ATP, and then that energy is then used in building up macromolecules from precursors. I hope that makes sense. These refer to acquiring hydrogen ions, and they'll move it to another reaction. So it just stores, and not only facilitates the initial reaction, it also facilitates subsequent reactions through their movement. I hope this makes sense. I have some of this is potential energy. Chemical bonds equal potential energy. Kinetic energy is basically everything else. So that's about the extent you need to know. I've color-coded it if you need. And you can see how these are reduced and then oxidized and then reduced and oxidized kind of to create energy, reduced to store energy. I hope that makes sense. Coupling refers to using energy release in exergonic catabolic reactions to supply free energy in anabolic reactions. ATP is an immediate in this process, and you'll have ADP being converted to ATP with free energy release in a catabolic reaction, and this will have lots of chemical potential energy, and as ATP is an intermediate, it will be required in anabolism to successfully build macromolecules. This is a pretty self-evident. ATP is used to release energy. That's all it's the same. Energy is required to synthesize ATP. Exergonic reactions release energy, and this is used to create ATP. Endogenic reactions require energy, which are required from the ATP. Hydrolysis releases energy, creates ADP and a phosphate. We're almost done. I think 2 more slides. Enzyme interactions. There are things called prosthetic groups. I was confused about this. It's not super important, but I didn't see sources for this, so I thought I'd explain it. Prosthetic groups are coenzymes or metal ions that are very tightly bound or covalently bound to the enzyme. For example, iron and heme. It often doesn't get removed. An inorganic cofactor is can be reversible, and it's an example is magnesium in DNA polymerase. So a cofactor is different from a prosthetic group in that it isn't necessarily permanently bound. It's there to assist the reaction. A coenzyme, an example of coenzymes are basically all vitamins. Lots of vitamins derive into coenzymes, and coenzymes work by they really add or remove functional groups required to assist a reaction, and in doing so, they assist enzymes in making reactions. They're not permanently bound. It's very reversible. Important examples are NADH and FADH2. I hope that's clear as well. It's not super important, but it's good to know just for interest as well. Enzyme regulation. There's really 2 primarily forms. One is gene expression. We've talked about enhancers and repressors. So gene expression can enhance or diminish enzyme synthesis. It may also impair the function of the enzyme, so it is less effective in catalyzing a reaction, which may be structural. So essentially, I don't know, there's things called allosteric enzymes. So you can have allosteric binding that can kind of positively or negatively influence the production of more protein and more DNA synthesis. There's also physical enzyme inhibitors. There's 3 different types you should know, and I've added allosteric just for completeness. Competitive enzyme binds to an active site and prevents substrates from binding there themselves. An uncompetitive enzyme binds to an enzyme substrate and prevents the release of products. You can see that in C. You can see in B, competitive. It's not letting the green one go in. And finally, in D is non-competitive. A substance binds in a site that isn't the active site, and this changes the conformation of the enzyme so the substrate cannot enter. Just for completeness again, allosteric is a reversible non-covalent bonding to a site other than an active site. It changes the conformation of the protein and can enhance or impair the catalyzing effect of the enzyme. That's all for me. I'll give it off to Danny Dog. Thanks for answering the questions in the chat. I'll have a look at them now as well. So yeah, thanks, Andrew.
If the telomerase was not active in most human somatic cells, what happens when telomeres are completely reduced and the genes are affected? So that's kind of what happens when we age. It's basically when you age, like you start to lose your telomeres at the end of your genes, and then yeah, you literally just start losing your chromosomes basically. And different stuff like stress, whatever, can like disable your telomerase and all of that sort of. There's a lot of different factors actually affect the telomerase and like basically affects how you age. So yeah, ideally you'd want the telomerase to be on all the time, but it's not. And yeah, when it's not, we sort of get the shortening of telomeres, and that just happens as we age. It's just sort of how we, I don't know, slowly die, I guess. But
Yeah,
anyways,
just to add in really, really quick, there's this new area of science right now where they're trying to like reverse that shortening of the telomeres, specifically like for anti-aging therapies. But you're exactly right, exactly what Andrew was saying. If they get shorter and shorter, that's pretty much how biologically our cells start dying.
Yeah. So now I'll talk a bit more about fuels, but if you do have any more questions, just chuck them in and we'll try to answer all of them. We have tried to go like a bit more quickly this week, just like not take up too much time. So hopefully it wasn't too confusing, but yeah, fuels. And next slide, please. So in your metabolism to get energy, your lectures will sort of talk about getting energy from three main fuels. So you have your carbohydrates, which are made of carbon, hydrogen, oxygen, and you have your lipids, which are like long carbon chains basically, and then your proteins, which have like an amino group and like a little carbon in the center. But yeah, so mostly you'll talk about carbohydrates and lipids. You won't talk about proteins as much for fuels, I don't believe. Next slide, please. So with glucose, you can get them from, well, mainly two sources, either from your meals or from glycogen, which is basically a bunch of glucose. So your glycogen would come from previous meals, which you had extra glucose, and then you turn that into glycogen. And then a bit later, when you haven't eaten for a while, they'll turn the glycogen back into glucose to use. So, but to first get glucose, you can hydrolyze carbohydrates, which basically means break down carbohydrates from your food. And then this glucose will enter glycolysis and then the citric acid cycle and oxidative phosphorylation if you have enough oxygen. And we'll talk about this more later. But the first step is glycolysis, which is the first step of breaking it down. So it's 10 enzyme catalyzed reactions. There's 10 reactions basically, but you don't need to know all the reactions. So with all of these things, you'll have a lot of reactions, but you actually don't need to know basically any of them. You just need to know major points. So glycolysis, what's important is it occurs in the cytoplasm with or without oxygen. It doesn't care because it doesn't actually need oxygen at this stage. Oxygen is only really needed later on. So what it does is a glucose, 6 carbon, is made into 2 pyruvate molecules, which have 3 carbon. The first 5 steps, first 5 reactions are what are called preparatory. So step 1 and 3 actually use an ATP each, and the last 5 steps are called the payout phase, and 7 and 10 produce 2 ATP each. So you have a net gain of 2 ATP. And step 6 is where you produce NADH. So these are sort of the most important bits of glycolysis you need to remember. So this is like the diagram. Like I said, you don't really need to know the steps, but it's just out of interest if you would like to look at it yourself.
Andrew, can I just interrupt you for a second?
Yeah, sure.
Just on the chat, we asked another question about the telomeres. That's your lifespan. So telomeres aren't necessarily what kills you. If you die of old age, it's going to be from like maybe from organ failure or your organs not working as well. Telomeres are more associated to the aging process. So there's a bit of a differentiation there. But just because I'm sharing screen, I cannot respond to you in chat. So maybe someone else can start respond to you in chat. Sorry about that. Sorry about that, Andrew.
That's all right. Yeah. So yeah, it just sort of contributes to you aging, but it probably won't kill you. It'll probably be like, you know, a heart attack or like some other disease that It's not really directly from the telomeres being shortened. Yep, next slide. Okay, so what happens to pyruvate? Pyruvate can go into like two directions depending on whether you have enough or not enough oxygen. So if you do have enough oxygen, which is maybe if you're just sitting, then the link reaction occurs. So what happens is pyruvate is oxidized to acetyl-CoA. So you'll put in 2 pyruvate because you've got 2 pyruvate from 1 glucose, and 2 NAD plus is needed. And your outputs, what you will get 2 acetyl-CoA from the pyruvate and then 2 NADH, and you also release a bit of carbon dioxide. So yeah, inputs and outputs are kind of the most important things with these reactions and in general, like all of these biochemistry steps. And what's also important is the key enzyme. So you should always remember the key enzymes as well. So in this case, it's pyruvate dehydrogenase, and this actually needs something called thiamine, which is a group B vitamin. I think that got cut off by the picture. I'm sorry about that. So it's a group B vitamin, and it does come up in questions sometimes. So if there's something wrong with the link reaction, sometimes it'll be that the patient doesn't have enough thiamine and that you need to give them more thiamine. And yeah, this occurs in the mitochondrial matrix as opposed to the cytoplasm as before. And what happens when you have insufficient oxygen? So perhaps, when you're exercising, is that the pyruvate enters the Cori cycle. So it undergoes fermentation to form lactate, which is the same thing as lactic acid. It's just like a slightly different form, but they're basically the same thing. And you have 2 inputs, like the 2 pyruvate from before. If it didn't have enough oxygen, it would go into here, and then it would form 2 lactate and 2 NAD+. So the key enzyme here is lactate dehydrogenase, and this reaction kind of recycles NAD+, which is quite important. So we'll touch on this again later. But what happens is you can have the buildup of lactate, which will cause lactic acidosis. And this is treated by giving the patient some sodium bicarbonate, which is base. So, base acid cancels each other out. And yeah, as you exercise, you will build up more lactate, but you probably won't build up too much lactate that, you'll go to the hospital and you need sodium bicarbonate. But yeah, so the link reaction, minor point, more of a fast reaction compared to the Krebs citric acid cycle, which follows the link reaction, but not this reaction. And we'll talk about that now, I think, in the next slide. Yep, so another minor point, acetyl-CoA doesn't always have to go into the Krebs cycle. The Krebs cycle is what happens when your body wants to make energy, it goes in a Krebs cycle. But acetyl-CoA can also go elsewhere. So it might be used to synthesize fat, for example. It might be used to make fat. So it doesn't always have to go here, but yeah, well, this is what we're looking at the moment. And once again, you don't need to know all the steps, but you just need to know from 1 acetyl-CoA, you'll get 3 NADH, 1 FADH2, and 1 GT is equivalent to 1 ATP. And that also got cut off by the picture. I'm sorry about that. Next slide, please. So after the Krebs cycle comes oxidative phosphorylation, or the electron transport chain. So in the electron transport chain, we will have a diagram coming up soon. There are 4 protein complexes. Their names aren't too important, but it is good to know that cytochrome C is part of the chain. It's not actually one of the complexes itself, but it is involved in the chain. And what's important about cytochrome C, which you might have heard of, is that it's blocked by cyanide. So cyanide is an inhibitor. And so how cyanide actually kills you, which you might have heard from James Bond or something, is it blocks your cytochrome C, and that because your cytochrome C is blocked, you can't actually make any more energy. And without energy, obviously you're going to die. So you die from a lack of ATP, basically. So we've mentioned NADH and FADH2 quite a few times up to this point from each of these reactions. And this is where they really come into the picture. So what they do is they give the complexes their own electrons that they received from the earlier reactions. So NAD plus, doesn't doesn't have the electron, but NADH after the reaction, which is NADH is formed, that will have the electrons from the reaction. And they'll give the complexes their electrons. And as the electrons flow through these complexes, this actually gives the proteins energy to pump hydrogen ions. What's the difference between NADH and FADH2? So NADH, they're both electron carriers, but NADH carries more energy compared to FADH2. But for your purposes, just think of them as both electron carriers. The chemical structures, etc., aren't important. Faculty really just wants you to know that electron carriers. I haven't read the chat yet, but I'll have a look now. Is oxidative phosphorylation the set of reactions and electron transport chain, the combination of compounds? So oxidative phosphorylation kind of refers to this whole process. We'll talk a little bit more. I think I'll talk a little bit more about this later because it'll become a bit clearer. So just hold on for a sec. But yeah, continuing on, electrons flowing from the complexes give the energy to pump hydrogen ions from the mitochondrial matrix into the intermembrane space of the mitochondria. So mitochondria have two membranes. I don't know if you know that. Yeah, so it pumps H+ from the matrix into the intermembrane space. And then finally, the electron flows from complex 4 to react with oxygen. And this is why you need oxygen. This is where oxygen is important. It's not used before, but this is where it's important, and it forms water. And that's how you get rid of the electrons. And then ATP synthase then allows the H+ to kind of flow back into the matrix from the intermembrane space to produce ATP. So the next slide might make everything a bit clearer if it's a little bit confusing right now. So this is a good diagram, I think. So here are all of your complexes, 1, 2, 3, and 4. Oh, I can't draw 4, but yeah. So these are all of your complexes, and you can see that NADH and FADH2, they come up and then they give an electron and they become NAD+ again. FADH2 gives an electron, becomes FAD again, which is what they were before we had the previous reactions. So the previous reactions are basically to form NADH and FADH2 to give the electrons to these proteins, basically. That's the whole thing we've been trying to do this whole time. And they'll pump up H+ ions through 1, 3, and 4. You'll notice that a complex 2 doesn't actually pump any. But yes, this is the intermembrane space. So this is the outer membrane, and then this is like the inner membrane. So here is the matrix, here is the intermembrane space, and here is the cytoplasm, which is outside of the mitochondria. It pumps the H+ into the intermembrane space, and that you have your ATP synthase here, which is quite important and is the key to all of this. And it will let H+ flow back through itself. And in the process, it will use like this passive flow to generate power. So it'll turn ADP into ATP. And obviously ATP, is energy. And you should know that there's a high concentration of H+ ions in the intermembrane space because of all the pumping of the complexes. And then it flows through ATP synthase because of this high concentration gradient flowing into this low concentration. And to go back to the oxidative phosphorylation and electron transport chain question, the electron transport chain really just refers to this 4 complexes and the flow of these electrons. That's electron transport chain. And then this oxidative phosphorylation sort of more refers to the process of the ATP synthase, because to go from ADP to ATP, what you're actually doing is you're phosphorylating ADP to you're giving another phosphate atom basically, and this third phosphate atom will form a chemical bond, right, to the ADP to form ATP. This chemical bond is where you store the energy. So oxidative phosphorylation kind of refers to phosphate that you're sort of giving to this ADP. And it's oxidative because all of these reactions from beforehand were oxidative reactions. So it's oxidative phosphorylation. I hope that makes a bit of sense. If it doesn't make sense, let me know and I'll try to explain it again. And next slide, please, David. Oh, I should clear all these drawings. Annotate. How do I clear? How do I clear this? It's not letting me clear.
So go to annotate, then hover over clear, and then it'll just say clear all drawings.
I actually can't see it clear all drawings. Oh God, so boomer. Maybe you do. You have to clear it. I think you can clear it as the host, David.
Gotcha.
Yeah, thank you. So yeah, the core concepts that we've been saying so far is that NADH and FADH2, which you've got from the link reaction and the Krebs cycle, they're all basically electron carriers. So basically all of these reactions were to make these electron carriers. And then they can donate their electrons at the electron transport chain to produce ATP. That is the main point. So yeah, to repeat it once more, all the reactions in glycolysis, the link reaction, Krebs cycle are all in order to produce NADH and FADH2. You get a little bit of ATP from these reactions as well, but they're really nothing compared to how much you generate at the electron transport chain. That's where you get the bulk of your energy, and that's why it's basically like the main point. Can we go into the next slide, please? Just some numbers to remember. So in the ETC, electron transport chain, every 4H plus that flows through ATP synthase has enough energy to generate 1 ATP. So 1 NADH, its electron carries enough energy to cause 10 H plus ions to be pumped through, and FADH2 has enough energy to cause 6 H plus ions to be pumped through. So the maths comes down to it that 1 NADH generates 2.5 ATP, and 1 FADH2 generates 1.5 ATP. Obviously, you can't have half an ATP, so the bit of energy would carry over a bit later, and then, so if that sort of makes sense, it'll just carry later, and you'll be able to generate more ATP. When pumping up the H plus to intermembrane space, do complex 1 to 4 use energy? Yep, so complex 1 to 4 get the energy from the electrons that were donated. The electrons that were donated provide the energy for complex 1 to 4 to pump the H plus ions into the intermembrane space.
Just with that as well, if you think about it, H plus is positively charged, electrons are negatively charged. So there is already a spontaneous attraction between the two that facilitates H plus going up.
Yep, next slide, please. Another scenario that's quite important is what happens where there isn't enough oxygen. So you remember that oxygen we need at the end of the electron transport chain to react with O2 and form water. So if you have a buildup of electrons from not having enough oxygen to react with them and form water, then you'll have a buildup of electrons in the electron transport chain, and then your NADH and FADH2 cannot donate electrons because, well, the complexes already have electrons and it's all piled up. And as a result, because you can't donate the electrons from NADH and FADH2, there will be a buildup of NADH. So it's all kind of backlogging, all piling up. And if you have a buildup of NADH, then you're not forming any more NAD+. And you have used up all the NAD+ in the previous reactions, in the link reaction and the Krebs cycle, and you won't have any of it anymore. But NAD+ is really important for glycolysis, the link reaction, and the Krebs cycle. So you really need it for these reactions to go through and continue. So it's really important that you get some NAD+ back. So this is where the Cori cycle, instead of the link reaction, is favored, and you'll produce lactate instead. If you remember from the fermentation or the Cori cycle, the pyruvate, it forms lactate instead. And in the process, you use one NADH and you make one NAD+. So this is what it means when you sort of recycle the NADH. And once you have NAD+ back again from the Cori cycle, you can let glycolysis, the link reaction, everything continue again. And this is what sort of happens in exercise when you don't have enough oxygen. Your body will be like, oh, there's not enough oxygen. We need more NAD+. How are we going to get NAD+? We'll have to use the Cori cycle. And then because you're using the Cori cycle, you get lactate, and then you get, like your muscles become acidic because they're making lactate, and this is where you get your soreness and tiredness when you're exercising. And if you could continue, David. Pentose phosphate pathway. This thing's, you just need to know this exists. It basically never shows up, but it's just, you just need to know this exists. You don't need to know the details, you just need to know it exists. It's a parallel pathway to glycolysis. So instead of going through glycolysis, what can happen is that your glucose, it will be taken away and put into the pentose phosphate pathway. And it will generate NADPH and ribose 5-phosphate instead. NADPH is basically the same thing as NADH and FADH2 in the sense that it is just an electron carrier and it can donate its electrons in the electron transport chain. Ribose 5-phosphate, it goes to synthesize nucleotides, so like your DNA, etc. And that's really all you need to know about the pentose phosphate pathway. I don't even think they lecture about it, but it's just good to know that it exists. And lipid metabolism. So fatty acids are basically long carbon chains and they're more energy dense because they're more reduced. That means they can be oxidized more. And if you're oxidizing them more, you can make more electron carriers and that will produce more energy. So that's what it means when they are more energy dense. So glucose and glycogen are more short-term energy needs and quick delivery. So they're the stuff you use if you go for a sprint. Fats are longer term energy needs and are more slow delivery. So fats are the stuff you'd be using if you're just sitting sort of, and you had lunch a few hours ago, it's almost dinner, you're going to be using your fats. So you'll cover more of lipid metabolism and transportation next week. And you get lipids from your diet. So you eat some fat, like animal fat, whatever, oil, they'll get emulsified, which basically means, emulsified basically is like two liquids that don't mixing into one. And we say emulsified because fat is non-polar, right? And well, your stomach acid and all the fluids like water is polar. Non-polar and polar don't mix. But if you emulsify it means like you mix them, and how you emulsify is basically you release some stuff in your digestion and it breaks up the fat into like tiny chunks, and that allows them to mix basically. Not too important, but just if you were wondering. And then the fat is absorbed by your guts, the lipids, and then in your gut cells basically they are resynthesized into triglycerides, which are, if you haven't learned about them in high school chemistry, I think you would have, but if you haven't, they're basically just bigger fats, I guess. They're like 3 fatty acid chains attached to, they're basically 3 fatty acid chains attached together. And then what happens to these triglycerides is that they're packaged into what are called chylomicrons. So if you look at the bottom, that is a chylomicron. Inside the chylomicron, you have your lipid core, and that's where all the triglycerides are. And you can see that it's like 3 kind of squiggly things attached to a head. So that's like 3 fatty acid chains. The triglycerides are stored inside and they're surrounded by a surface coat. The surface coat is made of phospholipids and cholesterol and apolipoproteins. And you'll learn more about them next week. Why do you have to store them in this like sphere or this chylomicron? Is because fatty triglycerides, they're non-polar, so they can't dissolve in your blood. How are you going to transport them around if they can't dissolve in your blood? You need these phospholipids. Phospholipids, they have a polar head, and then they have these non-polar tails. And so if you have a polar head, then that can dissolve in your blood, and then you can transport all your triglycerides. Next slide, please. So the chylomicrons, they're first absorbed into your lymphatics, which I'm not sure if you've learned about them yet, but they're involved with your immune system, and they're kind of like your blood vessels in that they sort of link the whole body and they're everywhere. So first they absorb it into lymphatics, and then they enter your blood vessels and the systemic circulation. cells will have lipoprotein lipases on their surface, and they can break down the chylomicron to produce amino acids, fatty acids, and cholesterol to be absorbed. Fat cells are activated by glucagon, which is a hormone that you release if you have a low blood sugar. And this stimulates hormone-sensitive lipases on the fat cells to break down triglycerides, or TAGs, shortened, into fatty acids and glycerol. The fatty acids attach to albumin to move around in the circulation. Albumin is like a protein you have in your blood. Then they need to attach here because they're non-polar and they can't dissolve in the blood. And that basically moves around your whole body and delivers it to all of your cells. The glycerol is sent to your liver, and the glycerol can be processed by the liver to make fatty acids again. Next slide, please. Thanks. Next thing is beta oxidation. So beta oxidation is oxidation of fatty acid chains into acetyl-CoA. So you make acetyl-CoA from fatty acid chains. The fatty acid chains are first shuttled into the mitochondria by a molecule called carnitine. They can't diffuse across because they're too big. And so what happens when you have carnitine attached to it is then you have proteins that are in the membrane that can attach to this carnitine and basically pull it across. And that will get you into the mitochondrial matrix. So the reaction is like a cycle, it's cyclical, and 2 carbons are broken off in each cycle. So if you have an 18 carbon long fatty acid, you'll break off 2 carbon atoms each time. You'll go from 18 to 16 to 14 to 12, all the way until it's broken into 2s, into all acetyl-CoAs. And each time you go through one of these breakages or cycles, you'll produce 1 acetyl-CoA from the 2 carbons that were broken off, 1 FADH and FADH2. So we have an example here. Beta oxidation of an 18 carbon long fatty acid. And something to note is faculty won't give you odd number ones. They can also be oxidized too, but the process is slightly different and faculty doesn't go into it. So they'll only give you like even numbered fatty acids. And if you have 18 carbon long fatty acid, it will take 8 cycles. Or you could just do like n divided by 2, take 1. If it confuses you a bit why you don't just divide by 2, just think about 4 carbon fatty acid chains. 4 divided by 2 is 2. Obviously, you only need to break it off once because, well, you know, if you break off 2 carbon, then you're left with 2 carbon. So you'll have 2 acetyl-CoA and you don't need to do it twice. So another thing to note is that 2 ATP is required to start the reaction. Next slide, please. So if you had to do 8 cycles, that means you'll end up with 9 acetyl-CoA and 8 NADH and 8 FADH2. 9 acetyl-CoA will then go to the Krebs cycle, and in the Krebs cycle, it will make 9 GTP, which is basically the same thing as ATP, 9 FADH2, and 27 NADH. So in total, you have 9 GTP, 35 NADH, and 17 FADH2 if you add up everything. So if you do the math, if you remember, 1 NADH is worth 2.5 ATP in the electron transport chain. You have 17 FADH2, which are worth 1.5 ATP in the electron transport chain, and then 9 GTP from the Krebs cycle. You minus 2 because you needed the 2 to start beta oxidation, and that will give you 120 net ADP. So you can see that fatty acids are very energy dense. Next slide, please. And yeah, that's the end actually. So I'll hand you guys over.
Thanks, Andrew. Yeah, so I'll be going over physiology, and for week 2, you guys are covering homeostasis and pH. So this is a nice and quick topic, so we'll get through it. Next slide, please. Yeah, so basically in high school chemistry, you would have learned that neutral pH is 7. And just a refresher of what pH is. So it's basically taking the negative log of your concentration of hydrogen ions. So the more hydrogen ions you have, the lower your pH is. So turns out that neutral pH is actually temperature dependent. So when you increase the temperature to 37 degrees Celsius, which is our body temperature, neutral pH is actually slightly lower. So it's going to be 6.8 in that case. So that's an important distinction to make. Now, when it comes to our blood, right, the normal pH of our blood is around 7.4, plus or minus 0.05. To determine whether it's alkaline or acidic, we need to compare it to the neutral pH at 37 degrees Celsius, because that's the temperature that our blood is going to be at. So 7.4 is higher than 6.8. So because of that, our blood is alkaline, even at a higher temperature, basically. Now, there's two terms, acidosis and alkalosis. These can be a bit confusing to wrap your head around. So basically, acidosis is when your blood pH is less than 7.4, and alkalosis is when your blood pH is greater than 7.4. So note that, let's say your blood pH, right, is 7.0. So it'd be acidosis, but technically it'd still be alkaline, as 7.0 is still higher than 6.8. So the important point is that acidosis and alkalosis, they're not determined relative to neutral pH, but rather what normal pH is. So if your pH is lower than normal, we call it acidosis, higher than normal, alkalosis. And then as to some things that can actually influence your blood pH, we have carbon dioxide. So here we have an equilibrium equation. So basically, when carbon dioxide is dissolved in water, so when you breathe in carbon dioxide, it's going to be dissolved within your blood. It's going to dissociate into bicarbonate and also a hydrogen ion. So basically, carbon dioxide is a source of acidity. So where this is sort of relevant, for example, if you increase your breathing or respiration rate, you're sort of removing more CO2 from your body, right? So because there's less CO2, there's going to be less hydrogen ions that dissociate. So you're going to have a decrease in hydrogen ion concentration. So you're going to become less acidic. Whereas if you sort of have a decrease in your respiratory rate, well, then CO2 is going to build up in your body. That's going to impact your blood pH. So we're going to have a increase in hydrogen ions, which is going to lead to a decrease in your pH. So you're going to get acidosis. But yeah, so pretty short for physiology this week, just those two main concepts.
Okay, now a little bit of HKS. There isn't that much in this week, so I'll kind of quickly go through it. Next slide, please. All right, so there's this thing called 4S, or twigs, and that is describing the 4S that we kind of describe a health system by, or twigs is the same thing. So the first S is stuff. So in a hospital, we need technology, the MRI, X-ray, different equipment to correctly provide care. So that's one thing. The second thing is we need staff. We need the GP, the nurses, receptionists, specialists, all these people at the hospital actually treating the patients. And that's the staff. And then there's also spaces. Obviously, we need hospital beds. We also need this radiology. We need pathology clinics, even the ED. We need all of these spaces, the infrastructure, to make sure that we are correctly giving care. And lastly, system. So this is kind of more tricky, and we'll cover a bit more later, but this is kind of like government and just other ways of connecting different components to each other within the hospitals and outside. So next slide. Just a quick note on rural areas. I believe you could get taught about this more in SEM 2, and you do assignment on it as well. Basically, the general gist is that there's lower numbers of doctors in rural areas compared to the major cities, and that's represented in this graph. Got straight from the Australian government page, so it's pretty accurate. Yep, so next slide. There's three things that a national health system should do: regulate, fund, and deliver. And we'll talk about how they should do those. And next slide, please. So the Australian healthcare system, how they regulate, is actually dependent on these four different things. So we obviously, in Australia, we have a federal government and a state government and a local government, but local doesn't really take that much responsibility in healthcare. But in terms of federal government, the federal government makes these type of national policies, is responsible for Medicare and also the pharmaceutical benefit scheme, the PBS, and other things, research funding, but kind of like the big dog. So that's kind of like what they're responsible for. Now, the state government is responsible for the public hospital management, like even the ambulances and the health complaints, which you hopefully already covered in a little bit. Actually, maybe not yet, but you cover it in ethics and medical law. And it's basically, you can complain about, one of the treatments you might have gotten from a doctor, and the state government will take care of that, not the federal government. Also, the immunization services, although I believe for coronavirus, it might be a bit different, but mostly for other immunizations, normally the state government takes care of that. And then there's the private part, which is just simply the private health insurance, like Bupa. And these also kind of take into consideration the medications not under the PBS, so not under the federal government things, and also private hospitals. And then the non-government or shared components are the pharmaceutical regulation and education. That's Aboriginal Torres Strait Islander funding, quality control, health workforce. That's a kind of like either shared or not really covered by the government. Next slide. All right, so there's also funding, which kind of mentioned before, and there's three different models that we normally talk about for a healthcare system, national healthcare system. First one is welfare state model, so that's funded completely by tax dollars, meaning the government has full responsibility. The difference we'll talk about a bit later, Medicare and PBS. So welfare is just completely run by the government, basically. So there's really no private insurance or stuff like that. It's really rare to see a country with a national health system with just based on welfare state model. Then there's the market model, which is basically the government has no responsibility, takes no real actions towards healthcare, and instead all the individual citizens and institutions basically make their own choices and pay for it. So it's kind of like US, it's not exactly, but like US, it's really, really driven on just money, basically. So there might be a video that you guys will assign to watch, but it shows you how US, despite spending so much money per year on hospitals, they have one of the worst hospital rankings among pretty good countries. So yeah, it's not great, but that's a market model. And then there's the hybrid model, which is the one that we use. And it's obviously a mixture, as it's suggesting the name, and the government provides public systems for basic coverage. So you can go to a public hospital and get basic coverage if you need. But then if individuals do want more coverage on top of that, they can get private healthcare. And that's exactly what we do in Australia, and it's one of the best in the world. So next slide. So yeah, this is Medicare that we were talking about before. And what Medicare is, it's a publicly funded universal healthcare insurance scheme in Australia, and it's operated by the Commonwealth or the Australian government. It's straight from Wikipedia, but that's what it is. So it's basically publicly funded and funded to help people in Australia, and it's run by the government. And what they basically do is they have two components. One is getting free treatment at public hospitals. So at public hospitals, you get free treatment under Medicare. And secondly, you can also get free or subsidized treatment from anyone with a Medicare provider number. So most GPs, even some specialists, some allied health, maybe even physios, things like that. Actually, not maybe not physios, but basically anyone with Medicare provider number, you can get not always free, but definitely cheaper treatment than if you did not have Medicare. So that's what Medicare is, kind of to help you with basically paying if you are an Australian citizen, which we'll talk about. And this Medicare levy, I believe, is not that important, but it's kind of just something that we will talk about. And it's describing how Medicare is funded. So it's funded through general tax, 2% of taxable income. And then there's a further surcharge levy up to 1.5% taxable income. And that's for those who are not on private health insurance. And 25% of the Commonwealth government's total health funding is gone towards Medicare. So it's quite a lot of money gone to Medicare. So you went back. Next slide. So now this is PBS. So PBS is basically also, again, a government funded service, but it's basically to make sure that the drugs or medications, as they say, medications are affordable for everyone. Well, not everyone, but affordable. So it's not too high. And also make sure that the safety and quality of drugs are actually to the standard that we want. So that's obviously down, you can see the difference, which is that Medicare is just free treatment, whereas PBS is kind of just making sure that the drugs that we allow within Australia are affordable and safe and the quality is good. How does it work then? Well, 87% of it is covered or paid by the government, and 13% is covered by us. And there's also other things like concession, where you get extra amount of, and also DVA or seniors card, threshold, PBS safety net. It's just talking about how if we have too much of people buying a certain drug, we might need to increase the price as a safety net. Now, next slide, I believe this is the last slide. Yeah. And lastly, this is just types of care. So there's 3 types: primary, secondary, and tertiary. Primary is like the first point of contact, and that's normally a GP, a nurse at a hospital, or an ambulance if you're going towards an ED. Then the secondary type of care is the specialist care. So it may be the GP refers you to a specialist for some reason, or an acute care in ED. So after you've been to an ambulance, you go to ED. That's secondary, as it kind of makes sense because you kind of go through one person. Now, tertiary is going through the secondary type of care already. So that's normally consultative specialist care. And normally it's when you are already in a hospital. So when you're already in the hospital, and for example, you've been here for a while and it doesn't seem to recover, they might need to refer you to another specialist within the hospital. That will be tertiary care. And yeah, I believe that's it. If you go next slide. Yeah, I believe that's it. So thank you guys. Any questions?